Alright, ji, assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome or welcome back to my YouTube channel. So in today's video, we will be looking at ASN A-Level Biology 9700 paper 1, and specifically we are going to discuss May-June 2025 paper 1 to over here, and this paper is of 1 hour and 15 minutes. So let's start with question number 1. Now question number 1 over here says that which row shows the possible uses of the eyepiece graticule. So what is the eyepiece graticule? Eyepiece graticule is basically a small scale inside the eyepiece of a microscope. Now they are saying that which row shows the possible uses. So we have to see the correct uses for the eyepiece graticule. So calibrating a stage micrometer viewed with a 10x objective lens. So this statement is incorrect. Why this statement is incorrect? Because we don't calibrate the stage micrometer, but instead we use the stage micrometer to calibrate the eyepiece graticule. Right? We use the stage micrometer to calibrate the eyepiece graticule. Right? For example, over here you can see that this is the stage micrometer scale, right? And we are using this stage micrometer scale to calibrate the eyepiece graticule. So basically we use the stage micrometer scale to calibrate the eyepiece graticule. So, and over here in this option they are saying that we are using the eyepiece graticule to calibrate the stage micrometer. So this is incorrect, right? And then the another statement which is given over here is this, that comparing the diameter of two cells viewed with a 40x objective lens. Yes, this is possible because once it is calibrated, once the eyepiece graticule is calibrated, so it can be used to measure and compare the cell sizes. So this is correct, right? So hence the correct answer for this MCQ would be option B over here. So I hope that this MCQ is clear to all of you. Let's now move on to question number 2 over here, which says that which statement about the light microscope is correct. Statement A is saying that as the distance to see 2 points as separate points decreases, the resolution also decreases. So no, this statement is incorrect. Because why? Because actually if you can distinguish the 2 points at a smaller distance, for example the light microscope is having the limit of resolution as 200 nanometers, and the electron microscope is having the limit of resolution as 0.5 nanometers. So if these objects, if these 2 points are apart till 0.5 nanometers apart, and you still can view them as the 2 separate points, then you will say that and the resolution is better, it is increasing. That's why we say that the electron microscope has a higher resolution as compared to the you can say the light microscope, because we are able to see the 2 points as the separate point, right? So as the distance to see the 2 points as the separate point decreases, the resolution increases, right? The resolution increases. Then they are saying that a bacterium 0.2 micrometer in diameter will not be visible if the resolution is 200 nanometer. If they are saying that the limit of resolution for the light microscope is 220 nanometers, right? And if it is 220 nanometers, so if we convert it into the micrometer, so we divide this value by 1000, right? And if we divide this value by 1000, so it will be 0.220. So if the resolution is 0.220 micrometers, so if the distance, if the diameters, so this is the limit of the resolution over here, and the bacteria is actually 0.2 micrometer, which is lesser than the limit of resolution, lesser than the limit of resolution. If the diameter of the bacteria is lesser than the limit of the resolution, then it will not be visible, right? It will not be visible. If it would be 0.22 micrometer, or it would be greater than 0.22 micrometer, then it would be visible, but because it is lesser than the limit of resolution, which is 200 nanometer, converted it is 0.22 micrometer, which is less than which is greater than the diameter of the bacteria, which is 0.2 micrometers, so it will not be visible. So this statement is actually correct over here. So we will say that this statement is correct. So option B is our correct answer. But we will also see other options as well, that why the other options are not correct. Two membranes that are less than 300 nanometers apart will be visible as two separate membranes if the wavelength of the light is 600 nanometers. So if the wavelength of the light is 600 nanometers, so what will be the limit of resolution? Limit of resolution is basically the least distance at which the 2 points can be paired as a separate point. So to calculate the limit of resolution, we will divide the wavelength by 2, so we will get 300. So if the object, if the membranes are 300 nanometer apart, or they are greater than 300 nanometers apart, then they can be seen as the separate membranes. But if it is lesser than 300 nanometer, then they cannot be visible as the two separate membrane. It will be visible as one point, right? So this is incorrect. The resolution will improve with visible light of a longer wavelength, such as red light. So no, this statement is also incorrect. Why this statement is incorrect? Because we studied that the shorter length gives you the shorter wavelength basically gives you the better resolution. The smaller the wavelength, the better will be the resolution. Like for example, over here there was the wavelength of 600 nanometers, right? And then if I say that there is a wavelength of 300 nanometers, and if we calculate the limit of resolution for a shorter wavelength, we are going to get 150 nanometers. Means that the 2 points that are 150 nanometers apart or more than 150 nanometers apart can be paired as the two separate points, right? So the better resolution is with the shorter wavelength. So we can say that this statement is also incorrect. So hence the correct answer over here is option B. I hope that this is clear to all of you. Alright, so now let's move on to question number 3, all of you. Question number 3 over here says that the electron micrograph shows a cell. What is the actual maximum length of the cell? So basically they have given us the magnification over here, and in case you guys are wondering where is the cell, so this is the cell over here, and this is most probably the nucleus of the cell. They are asking us to calculate the actual maximum length of the cell. So what is the formula for the magnification? Magnification is equal to image length divided by the actual length. So if you guys see over here, they are saying actual maximum length over here. So in order to calculate the actual maximum length, we must take the image length at its maximum, right? We know that the magnification is 108000, so we are going to put the magnification as it, right? And then what we are going to do is that we are going to calculate the image length. How are we going to calculate the image length? We are going to use the ruler. Over here I will just tell you the technique which you will use in order to calculate the image length, because obviously I am solving it online and I am solving it in a soft copy, right? So my measurements will be different, and the measurements which will be there on an A4 size paper will be different, and then the measurements of the picture on the actual CAI paper will be different. So it's better for you guys to understand the concept and then just apply your knowledge to solve it. So the maximum length would be somewhere over here, right? Somewhere this is somewhat the maximum length, which is coming to be about 6.5678. Let's suppose it is 6.8 over here, right? Let's suppose that the image length that is coming, the maximum image length that is coming is 6.8 centimeter. Now we are going to convert this centimeter into millimeter, and we will convert this centimeter into millimeter by multiplying it by 10, right? So this will be 68 millimeter, but because the value of the maximum actual length is given in micrometer, but because the actual maximum length in the answer is given to be in the micrometer, so you are going to convert this millimeter into micrometer by multiplying 68 by 1000, right? So you will add 68000 as the image length, right? And then the magnification you will write over here, and you will make the actual length the subject, and then you are going to divide these values, whatever the answer you are getting. Let me try it out if I am getting the answer similar. My answer is coming to be about 0.629, right, which is most closely over here 0.676, right? So the answer in the marking scheme was also option B, so it is somewhat closely coming to be around this, so hence we will consider option B as our correct answer. I hope that you got the way and you got the technique that how are we actually going to solve this question. Coming to question number 4, all of you, the question number 4 over here says that the table compares some biochemical molecules of P, Q, R and S. Now the biochemical molecules they have given us is the chlorophyll, DNA, RNA and the peptidoglycan. The tick over here basically represents that these are present in P, Q, R and S, and the cross represents that it is not present, right? Now further in the question they are saying that which row correctly identifies P, Q, R and S. So basically they have given us the chloroplast, virus, mitochondrion and the bacteria. So we have to basically select that what is P, Q, R and S. So let's discuss this, that the chlorophyll basically is present. Now if the chlorophyll is present, so we know that it can be the chloroplast. We can clearly tell that the chloroplast will be there where the chlorophyll is present, because obviously the chlorophyll cannot be present in the virus, or the chlorophyll cannot be present in the mitochondria, the chlorophyll cannot be present in the bacteria. So the option where the chlorophyll will be present would be the chloroplast. So we can clearly say that yes, the P is the chloroplast, because yes, the DNA is also present in the chloroplast, RNA is also present in the chloroplast, and we know that the peptidoglycan is not present in the chloroplast. Peptidoglycan is the component of the cell wall of bacterial cell. So I think I have also found for the bacteria as well that this would be the bacteria. R would be the bacteria. Why? Because peptidoglycan is a cell component of the bacterial cell wall, and only in the P, only in the R, the peptidoglycan is present. So R is the bacteria, right? Now virus, if I talk about the virus, so virus would be the S most probably. Why? Because viruses have a very simple structure. They either contain DNA or they either contain RNA. They do not contain both. So if you see over here in the S, RNA is thick and the DNA is cross, and chlorophyll is also cross and the peptidoglycan is also cross, right? So they contain the DNA or RNA surrounded by the protein coat, right? So we will say that S is the virus, R is the bacterium, P is the chloroplast, and obviously the remaining Q will be the mitochondrion, because mitochondrion has a DNA, circular DNA, it has RNA, and it does not have the peptidoglycan. And mitochondrion is a double membrane bound organelle. So hence the correct answer over here would be option B. So I hope that this MCQ is clear to all of you. Now question number 5 over here says that which cell structures contain enzymes and are enclosed by a double membrane. So basically we have to look for the 2 pointers over here, contain the enzymes and they are enclosed by the double membrane as well. So first of all, if I talk about the nucleus over here, so nucleus, we know that nuclear envelope is there, and nuclear envelope is double membrane bound. Nuclear envelope is double membrane bound. Now do the nucleus have any enzymes? So yes, the nucleus is having the enzymes as well. Enzymes for the transcription, right? Transcription from the DNA when the mRNA is made. So a transcription enzyme is used, and then when the mRNA is converted into the DNA, then also the enzyme is used. So yes, the nucleus is having the enzymes as well. And then the mitochondrion. So we know that mitochondrion and the chloroplast, they both are the double membrane bound organelles and they both contain the enzymes, right? The mitochondrion also contain the enzyme, because we know that the mitochondrion is the site of the cellular respiration, right? Mitochondrion is the site of the cellular respiration. So we know that the mitochondrion is having the enzymes which take part in the respiration, and chloroplast is, we know that it is a site for the photosynthesis. It is the site for the photosynthesis, so it has the enzymes involved in the photosynthesis. So hence 1, 2 and 3, all of these three structures, all of these three cell organelles contain the enzyme as well as they are the double membrane bound organelles. So hence the correct answer is option A. Alright, so now let's move on to question number 6. Question number 6 over here says that what is the order of size of the cell structure. So they sometimes ask you the order of the size of the cell structures, the size of the cell organelles as well. So for your knowledge, I am telling you that the smallest one is the nucleoli, right? The smallest one is the nucleoli, and then there comes the lysosome. Then there comes the lysosome, then there comes the centriole, and then there comes the ribosome. So the smallest one over here is the ribosome and the largest one over here is the nucleoli. Nucleoli is the largest one over here. So the ribosomes are the smallest, and then there comes the centrioles, which are larger than the ribosomes but smaller than the lysosomes. And lysosomes are greater in size than the centriole, and finally the nucleoli is the largest of all, the ribosome, the centrioles and the lysosome as well. So hence the correct answer over here would be that the largest one would be the nucleoli and the smallest one would be the ribosomes. So the correct answer over here would be option C. I hope that this is clear to all of you. Also, if they have given us this question over here, then they can give us a similar question in October, November 2025 as well, but with the different organelles. So you must have a good knowledge about the size of the cell organelles, starting from the largest size to the smallest size, right? So let's discuss the cell organelles starting from the largest size to the smallest size. Let me open my lecture notes first of all. If you guys want to see this lecture or you want to take the videos of this lecture, you guys can contact me on the given WhatsApp number which is in the description of my channel link. So let's write it down over here that what are the size of the cell organelles starting from the largest organelle to the smallest organelle. Starting from the largest to the smallest. G. So सबसे पहले we will have the nucleus. Nucleus would be the largest of all of the organelles, right? And then there would come the nucleolus. Then there would come the mitochondria. Mitochondria is smaller than the nucleolus. And then there comes the chloroplast. Chloroplast, usually in the animal cells, the chloroplast will also be included in the animal cell, right? And then there comes the lysosome, then the centrioles, and then the smallest one comes the ribosomes. if in case you guys want to include the endoplasmic reticulum and the Golgi apparatus over here as well, so the endoplasmic reticulum and the Golgi apparatus both are lesser in size than the nucleolus but greater in size than the mitochondria, right? So after the nucleolus there will come the ER, which is the endoplasmic reticulum. And then there comes the Golgi apparatus. So this is the whole structure of, so this is the whole cell organelle part, starting from the largest organelle going all the way to the smallest organelle. Now let us move on to question number 7, all of you. Question number 7 over here says that a sample of solution of sucrose tested for the reducing sugars remain blue. But when the another sample of the same solution is tested for the non-reducing sugars, the solution turns red. What explains these results? Alright, so if you over here know that sucrose is a non-reducing sugar. Sucrose is a non-reducing sugar. And the non-reducing sugar shows a negative result with the reducing sugar test. In the reducing sugar test, what do we do? That in the reducing sugar test, we simply put the Benedict solution in a sugar solution and the color changes. But because sucrose is a non-reducing sugar, so when we will add the Benedict solution in it, so it will remain blue. Let me move on to my lecture notes for explaining you the test for the non-reducing sugar. So when there is a test for the non-reducing sugar, if we simply use the Benedict in it, so it will give no color change. Why? Because Benedict directly is used for the test of the reducing sugar. So when the Benedict is there when the Benedict is put in the non-reducing sugar, it shows no color change, it remains blue. It means that no reducing sugars are present over there, right? Then what happened that we do the non-reducing sugar test, and when we do the non-reducing sugar test, how basically the non-reducing sugar test is performed? सबसे पहले we are going to boil the solution with HCl, right? What we are going to do, first of all we are going to boil the solution with the HCl. Now what does boiling the solution with the HCl does? Now if we boil the solution with the HCl, it basically causes the sucrose to undergo hydrolysis. It causes the sucrose to undergo hydrolysis and break down into its monosaccharide, which are the fructose and the glucose. And we know that the fructose and the glucose are the reducing sugars, so they can in turn react with the Benedict solution, right? So the reducing sugar fructose and glucose are formed when we boil the solution with the HCl due to the hydrolysis of the sucrose. Then what happens is that we will neutralize the solution with the alkali. Why we are neutralizing the solution with the alkali? Because we know that the Benedict solution works the best in the alkaline condition, right? So that's why we are neutralizing it with alkali, and then we are going to repeat the Benedict test, and then the color change will be seen. So this is basically how you are going to do a test for the non-reducing sugar, right? So let's read the options one by one and let's try to determine the correct answer over here. During the non-reducing sugar test, an acid hydrolyzes the sucrose to glucose and fructose. So I think we have got our correct answer, that yes. What explains these results? That during the non-reducing sugar test, because of the acid hydrolysis, the sucrose is converted into glucose and fructose. That's why when it is tested for the non-reducing sugar, the solution turns red. So the correct answer over here would be option A. Let's read the other options as well. The reducing sugar test hydrolyzes monosaccharides to disaccharides. No, it is wrong, because sucrose, we know that the sucrose is a disaccharide itself, and the disaccharides are basically converted into monosaccharide in the non-reducing sugar test. Non-reducing sugar test. And then if I talk about option C, the reducing sugar test converts sucrose into glucose and the fructose. It is converting into glucose and fructose, but not in the reducing sugar test, in the non-reducing sugar test. During the non-reducing sugar test, sucrose molecules condenses into the polysaccharide. No, it gets hydrolyzed to its monosaccharides. So hence this is the explanation for question number 7. Now let's move on to the explanation for question 8 over here. Now let's start question number 8 over here. It says that which molecules contain at least 2 double bond. Now they are saying that we have to mark that which molecules contain at least 2 double bonds, and they have given us the 3 molecules. One is the saturated triglyceride, the other is the collagen, and the third one is the hemoglobin. So let's start with the collagen. So we know that the collagen is a protein, right? And if the collagen is a protein, so it must be made of a lot of amino acids, and we know that how the amino acids combine with each other. For example, if this is a one amino acid, it combines with the another amino acid via the peptide bond, right? Via the peptide bond. And what is a peptide bond? Peptide bond is C double bond O NH. And they are saying that which molecules contain at least 2 double bonds. So because a single collagen molecule is having hundreds of the amino acids combined together, so it will have hundreds of the peptide bonds as well. And if they are having hundreds of peptide bond, then there may be hundreds of the C double bond O, right? So we can say that yes, it is having the multiple, collagen is having the multiple C double bond O, right? At least 2 double bonds they are saying, but the collagen, because it is made up of a lot of amino acids, so it will be having more than 2 C double bond O, right? It will be having more double bonds than 2. So we can say that yes, the collagen is having at least 2 double bonds. So we will take it. And then similarly, if I talk about the hemoglobin over here, so we know that hemoglobin is also made up of the 4 polypeptide chains, right? 4 polypeptide chains. 2 are the alpha globin chains, 2 are the alpha globin chains, and the 2 are the beta globin chains, right? And it additionally contains the heme group as well. And because let's talk about the protein part, that because it is also having the protein part, and we know that proteins are made up of the multiple amino acids, and amino acids are connected to each other by the peptide bond, and peptide bond is having the double bond. So obviously it will be having a lot of double bonds. So yes, hemoglobin will also be considered the correct option, right? Because it is also having at least 2 double bond. It is having more than 2 double bond, but at least 2 double bond they are having it. Now let's talk about the saturated triglyceride over here. Now by the term saturated, you may be tricking that the saturated fatty acids are not the saturated triglycerides, or the word saturated itself is saying that it is not having carbon-carbon double bond, right? But they are not talking about the saturated fatty acids. They are talking about saturated triglycerides. Now we know that how the triglycerides are formed, or what is the structure of the triglyceride. For example, if I put it over here, let's for now put this over here, right? Let's put it on a cleaner space so that you guys can also see it properly. Alright, so for now let's put it over here. Yes, so we know that the triglyceride is made up of a glycerol molecule, right? Triglyceride is made up of a glycerol molecule, which is the alcohol, and 3 fatty acids, right? This is the fatty acid. Fatty acid is determined by the C double bond OH group over here, right? 3 fatty acids and the alcohols combine together to give the triglycerides. Now how the basically triglycerides are formed over here, we know that how the triglycerides are formed. The H from the alcoholic group and the OH from the carboxylic acid group, acid group from the fatty acids is removed, right? H from the alcohol and OH from the carboxylic acid group is removed, and the three molecules of the H2O water have been eliminated over here, and then the triglyceride is formed joining the alcohol glycerol part and the triglyceride part. So over here you guys can see that it is having more than two double bond, it is having three double bond over here. So we can say that yes, the saturated triglycerides are also having the more than two double bonds, right, or at least two double bond. So hence the correct answer over here would be option D, that yes, all of these three structure, be it collagen, be it hemoglobin, or be it saturated triglyceride, all are containing at least two double bonds. So I hope that this makes sense to all of you. Moving on to question number 9. Question number 9 says that amylose, amylopectin and glycogen are all polysaccharides. Enzyme X removes one maltose molecule at a time from the ends of the polysaccharide molecules by the hydrolysis of alpha 1,4 glycosidic bond. So basically they are in the question saying that there is an enzyme X which does the hydrolysis of the alpha 1,4 glycosidic bond. Which row shows how completely each of these molecules will be hydrolyzed by the enzyme X? Assume that each polysaccharide is composed of the same number of monomers before the hydrolysis begins. So they have given us three structures. One is the amylose, and then the other is the amylopectin, and the third one is the glycogen. And they are asking that which of these in a sequence is least completely hydrolyzed and what of them is most completely hydrolyzed. So if I start with the amylose over here, so we know that amylose is a linear chain and it only has the alpha 1,4 glycosidic bond. Whereas if I talk about the amylopectin over here, so amylopectin is branched as well. So at the branches it is forming the alpha 1,6 glycosidic bond, and otherwise it is in the linear, over here in the linear chain it is having alpha 1,4, and on the branches where it is branching it is having alpha 1,6. And if I talk about the glycogen, so similar is the case for the glycogen as it is for the amylopectin, but over here the glycogen is more branched than the amylopectin, which means that the glycogen has more alpha 1,6 glycosidic bond, and alpha 1,6 glycosidic bonds are formed at the branches. The linear structure is formed by the alpha 1,4 glycosidic bond. So the least commonly hydrolyzed would be the glycogen. Why it would be the least commonly hydrolyzed? Because only alpha 1,4 glycosidic bonds are being hydrolyzed, not the alpha 1,6 glycosidic bond. And why the glycogen is least completely hydrolyzed than the amylopectin? This is majorly because the glycogen is more branched than the amylopectin, right? Because amylopectin is less branched, so it will be having less alpha 1,6 glycosidic bond. So the glycogen will be the least hydrolyzed. The amylopectin would be the a little less hydrolyzed than the glycogen, right? And then the amylose, as because it is a linear chain, only it is having alpha 1,4 glycosidic bond, so it will be hydrolyzed completely. So hence the correct answer over here would be option D. Now let's go on and solve question number 10. Question number 10 over here says that which property of water enables sweating to be an efficient means of losing heat from the body? So it is a very popular statement that the sweating causes cooling, right? It is a very popular statement that sweating causes cooling. Now how basically the sweating causes cooling? This is because of the high latent heat of vaporization of the water. Now what does the high latent heat of vaporization of the water means? It means that the high means that the water absorbs a large amount of heat energy so that it can evaporate from the skin, right? From the when the water is on the skin, so it is basically absorbing a high amount of heat energy from the body so that it can evaporate from the skin. So that's why, because a lot of heat is taken away in breaking those bonds of the water and causing them to evaporate from the skin, so that is why the cooling effect is being seen in the sweating. So you can say that what property of the sweating is efficient means of losing heat from the body. So it is option A, that the latent heat of vaporization of the water is high. Now why other options are not correct, let's see that as well. The specific heat capacity of the water is high. This property of water is correct, that yes, the specific heat capacity of water is high, but it regulates the temperature. Not directly it is relevant to the sweating or the cooling mechanism, right? It helps regulate the temperature, but it is not directly relevant to sweating. Then in option C, they are saying that each water molecule can form a hydrogen bond with 4 water molecules. This statement is correct, but it may explain the other properties of the water as well. It is not directly related to the cooling. The force of cohesion and adhesion on the water molecules are very high. So this is correct, that yes, the force of cohesion and adhesion on water molecules are very high, but it is not relevant to the sweating. Okay, it is not relevant to the sweating. It may explain the water transport in the xylem, but it is not relevant to the sweating. So this is also eliminated. So that's why we have selected the best option A as our correct answer. Now let's move on to question number 11, all of you. Now the question number 11 over here says that which bonds are found in the levels of the structure of the protein molecule. So if I talk about the primary structure of the protein, so primary structure of the protein is simply the sequence of the amino acids. And if I talk about the secondary structure of the proteins, so secondary structure of the protein consists of the alpha helix and the beta pleated sheets, right? And if I talk about the quaternary structure of the hemoglobin, so quaternary structure of the hemoglobin is the interaction between the two or more polypeptide chains. Now they are basically asking us majorly about the bonds that are involved in all of these three structures. So we know that because it is a sequence of the amino acids, just so the amino acids will be linked together by each other by the peptide bond, right? The more specific term to be used is the peptide bond, and we know that peptide bond is a type of a covalent bond, right? But to be more specific in terms of the, as we are talking about the protein molecule, so to be more specific we will say that the peptide bond. Then if I talk about the secondary structure, so the alpha helix and the beta pleated sheets are mainly formed by the interactions because of the hydrogen bonds, right? Due to the hydrogen bonds. So over here we will say that the secondary structure is the hydrogen bond. It cannot be ionic bond, right? And also over here if we consider the primary structure to be covalent bond, because peptide bond is a covalent bond. And if I say that the secondary structure is by the hydrogen bond, so the quaternary structure, this option A will be incorrect, because the quaternary structure is not mainly involved by the peptide bond, right? Quaternary structure is stabilized by a lot of the other forces as well, which includes the other forces and the bonds as well, which includes the ionic bonds, right? It includes the hydrogen bonds, it includes the hydrophobic interactions, and sometimes the hydrophobic interactions, and sometimes the disulfide bonds as well. So the quaternary structure is not majorly stabilized by the peptide bond. So the quaternary structure majorly contains the ionic bond. So the correct answer over here would be option C. I hope that this MCQ is clear. Moving on to question number 12. Question number 12 over here says that prokaryotes and eukaryotes use the extracellular enzymes. Extracellular digestive enzymes are found in the external environment of some prokaryotes. Alright, some prokaryotes are digested by the hydrolytic enzymes during the phagocytosis by the eukaryotic cells. Which row shows the location where the extracellular enzymes are produced? So be it the extracellular enzymes of the prokaryotes, be it the extracellular enzymes of the eukaryotes, both are basically produced inside the cell, because we know that the enzymes are basically proteins, right? Enzymes are made up of the proteins, and all of the process of the protein synthesis is taking place in the organelles inside the cell. For example, in the ribosomes, the protein synthesis is being taking place, and then after the ribosomes it may go to the Golgi apparatus to be modified, right, and then it is secreted out of the cell. So the site of the production in both the prokaryotes and the eukaryotes, enzymes are produced inside the cell, right. So site of the production of the digestive enzymes used by the prokaryotes would be inside the cell. Site of production of the hydrolytic enzymes used by the eukaryote phagocytes are also inside the cell, right. So enzymes are produced inside the cell, and then they can move out of the cell by the exocytosis, right, but they are synthesized inside the cell. Now moving on to question 13, it says that amylase breaks down the starch molecules. So we know that the starch molecule is made up of the maltose. So when the starch molecule will be broken down, so the maltose will be given off, and we know that maltose is made up of the alpha glucose monomers, right. It is made up of the two molecules of the alpha glucose monomers. So they are saying that which substance will have the same number of molecules for the duration of this reaction. So if I start to discuss the options one by one, so let us look at the amylase over here. So amylase, because it is an enzyme, and we know that enzymes are they speed up the reaction, but they are not used up in the reaction. So obviously their number will stay the same. So its number will not change, or it will have the same number of molecules during the whole duration of this reaction. Whereas if I talk about the water, so water is basically used in the hydrolysis. When starch is converting into maltose in the presence of the enzyme amylase, it is a hydrolysis process, and water is also used in this hydrolysis process, right. And if I talk about the maltose over here, so maltose is also the product of the breakdown of the hydrolysis of the starch. So it is also increasing, right. It will also increase as the reaction will continue. As more starch is going to be breakdown, more maltose will be there, so it will increase. Amylose, and amylose, we know that amylose is the substrate. So as the more starch will be broken down by the enzyme, so amylose, as the amylose is acting as a substrate right over here, so the substrate will decrease in the concentration, and more of the product, which is the hydrolysis product of the starch, maltose, will form. So you can say that amylose acts as a substrate over here, so it is going to decrease. So the correct answer over here is A, amylase, which is an enzyme. It speeds the reaction, but it is not used up in the reaction, and so its number of the molecules stay constant throughout the duration of the reaction. Now let's move on to question number 14 over here, which says that which graphs could show the effect of pH on the rate of the enzyme catalyzed reaction. So they have given us the 4 graphs over here, and they are asking us that out of which of these 4 graphs is showing the effect of pH on the rate of the enzyme catalyzed reaction. So to answer this question, you must have a knowledge that enzyme works the best at its optimum pH. The pH at which the enzyme works its best is the optimum pH, where the enzyme works the fastest. So you must know and should have this knowledge that on the either side of the optimum pH, for example, if this is the optimum pH of the enzyme, right, so on the either side of the optimum pH, the activity of the enzyme will drop sharply. So in the graphs where you will see the sharp drop is basically the graph where it is showing the effect of pH on the rate of the enzyme catalyzed reaction. So on the either side of the optimum pH, there must be a sharp drop. right? So why is the sharp drop there? Because we know that because of the pH the hydrogen ions are there, right? And if there is a disturbance in the hydrogen ions, so the hydrogen bonds and the ionic bonds in the enzymes both are disturbed, right? And because we know that the enzyme is most probably a tertiary structure and a quaternary structure, so if the hydrogen and the ionic bonds are disturbed, so it may change the enzyme's active site and cause the denaturation of the enzyme, for which the rate of reaction would decrease. So the graph number 2 is a very popular graph for the pH. So we know that yes, 2 is a graph for the pH. And if I talk about graph 3 over here, so in graph 3 you can see that there is a slight inclination and then it is becoming constant, and this is not the property of a graph of the pH, right? In the 4 you guys can see that there is one optimum level and then there is a sharp decrease in the activity after that optimum level is replaced, right? After that optimum level the activity is decreasing sharply. So you can say that yes, 4 and 2 both are the graph for the pH. Now why one is not the graph of the pH? Because it is showing optimum activity at two different pH, which cannot be the case. So hence the correct answer over here would be option C. 2 and 4 graph are showing the effect of pH on the rate of the enzyme catalyzed reaction. Moving on to another question, which is question number 15 over here. It says that after a heart attack, the enzyme lactate dehydrogenase leaks into the blood plasma from the damaged heart muscles. So they are saying that whenever the heart muscles get damaged during an heart attack, so that releases the enzyme known as the lactate dehydrogenase, also commonly known as the LDH, right? And then further in the question they are saying that what is required to measure the activity of the lactate dehydrogenase activity in a sample of the blood plasma. So we know that to measure the activity of the enzyme, which is the lactate dehydrogenase, we can introduce the substrate for the lactate dehydrogenase in the blood plasma, right? And we can monitor the activity of the enzyme by it. Like when the enzyme will obviously act on its substrate, so we can monitor the product formation. So by this we can actually measure the activity of the enzyme product formation. Now let's read the options one by one. So sterilization of the blood plasma by heating. So this is incorrect. Why this is incorrect? Because if we are sterilizing the blood plasma by heating, so we know that heat may denature the enzyme, right? Heat will denature the enzyme, and if the enzyme is denatured, so we cannot monitor its activity, right? Incubation of sample with substrate for the lactate dehydrogenase. So yes, this is correct, because if we add a specific substrate to the plasma sample, for example you can add the lactate, so if the LDH will be present, so it will catalyze that reaction, and then we can measure the rate of the product formation, and that rate of the product formation will actually help us to measure the activity of the lactate dehydrogenase. And then the other option they have given us is that incubation of the sample with lactate dehydrogenase inhibitor. Now this is also incorrect, because if we are using an inhibitor, or if we are introducing an inhibitor in the plasma, so inhibitors, we know that it blocks the enzyme activity, or it can reduce the enzyme activity, right? So this would prevent the measurement of the activity of the lactate dehydrogenase enzyme or the LDH enzyme. So it won't be suitable to measure the activity of the enzyme, because obviously it is an inhibitor and it will block the activity of the enzyme. So hence the correct answer over here would be option C. I hope that this is clear to all of you. Moving on to question number 16, it says that which graph correctly shows the value of the Km and the Vmax. So they have given us the 4 graphs and they are saying that which is correctly showing us the Km and the Vmax. So if we see over here and if we try to identify the correct value of the Vmax and the Km as shown in the graph, so we can see over here that this over here is the Vmax. So almost in every diagram the Vmax is correct, right? In almost in every diagram the Vmax is correct. So Vmax, what is actually the Vmax? Vmax is basically the maximum rate of reaction of the enzyme. It basically shows us the maximum rate of reaction of the enzyme, right? And what is the Km? Km is basically the substrate concentration. It is the substrate concentration at half Vmax. At half Vmax. So for example, if this is the Vmax and this is for suppose half Vmax, so it is the substrate concentration at half Vmax. So this is over here they are showing that the Km is over here on the y-axis, right? But the Km should not be shown on the y-axis, it should be shown on the x-axis because it is the substrate concentration at half Vmax. And also it is saying that the concentration of the enzyme on the x-axis, so this is incorrect because Km on, also the D is also incorrect because Km is the substrate concentration at half Vmax, so it can be this or this. Now over here they are showing the Km at the y-axis, which is also not possible because y-axis is showing us the rate of reaction. So the Km is the substrate concentration at half Vmax. For example, if this is the Vmax and if we halve this value of the Vmax, so we are going to get the value of the Km somewhat over here. So Km will be the substrate concentration at half Vmax. So hence the correct answer over here for this MCQ would be option B. This graph is basically showing the correct depiction of the Km and the Vmax. Let's have a look at question number 17 over here. Question number 17 over here says that cell membranes become less fluid as the temperature decreases. So we know that as the temperature decreases, the cell membrane becomes too rigid. So bacteria and yeast cannot regulate their cell temperature. How do the bacteria and yeast maintain the fluidity of their cell membranes when the temperature decreases? Because we know that when the temperature decreases, the cell membrane, they are saying that it becomes less fluid and more rigid. So they have given us the 3 statements and they are saying that out of these 3 statements tell us that how the bacteria and the yeast cell maintain their fluidity. So let's give each statement a read one by one and try to answer our question. Statement 1 over here says that they increase the number of the unsaturated fatty acids in their phospholipid molecule. So yes, this is correct. Why this is correct? Because as the unsaturated fatty acids increases, so there is a kink. We know that there is a kink in the unsaturated fatty acid, right? Due to this kink, what happens is that it prevents the tight packaging of the phospholipids and increases the fluidity of the cell membrane at the low temperature. As I am repeating that due to this double bond, what happens that the kinks are introduced in the hydrocarbon chain of the fatty acid, due to which the fluidity increases. Then in statement 2 they are saying that they increase the number of saturated fatty acids in their phospholipid molecules. So this is incorrect because the saturated fatty acids are simply having the straight chain, and due to this straight chain they will pack tightly, so it will reduce or decrease the fluidity, right? So it is not useful. So this is incorrect. And then statement 3 over here says that they increase the number of the cholesterol molecules in the cell membrane. So we know that yes, the cholesterol basically regulates the fluidity of the cell membrane. So this statement is correct because at low temperature, what does the cholesterol do? That at the low temperature it prevents the phospholipid from packing too closely, and at high temperature what happens that it restricts the movement of the phospholipid too far to reduce the excess fluidity. So it basically regulates the you can say the fluidity of the cell surface membrane at both the high temperature as well as the low temperature. So the correct answer over here would be option A, 1 and 3 only. These two statements are, or these two methods are done by the bacteria and yeast to maintain the fluidity of their cell membrane when the temperature decreases. Let's now do question number 18 over here. Question number 18 over here says that samples X, Y and Z are epidermal tissues cut from an onion. Each epidermal tissue was immersed in one of the three different concentrations of a salt solution for 30 minutes. A student observed each tissue sample with a light microscope. Then the student estimated the concentration of each salt solution using a scale. The diagram shows the scale P, Q and R represents the estimated concentration of the three salt solution. For example, if this is the distilled water, right? And if this is the distilled water, so it is having the more of the water in it and less of the salt concentration in it. Then the R is having a lot of salt in it, like it is concentrated in salt, so it is having less water and more solute, so it is a hypertonic solution, right? It is a hypertonic solution. And also Q is somewhat in the intermediate hypertonic, right? If I further read the question, they say that the photomicrograph shows the appearance of the tissues in the sample X, Y and Z. So if you see over here, sample X and sample Y are showing the plasmolysis, right? How it is showing the plasmolysis or how it is showing the plasmolysed cells, because you guys can see over here that this is the cell wall, right? And you guys can see that, for suppose this is the cytoplasm, it is being pulled away from the cell wall, and over here a lot of it has been pulled away from the cell wall. So this is the initial stage, and this is the later stage of the plasmolysis or the plasmolysed cells. And if I talk about the sample Z over here, so in the sample Z you guys can see that the cells are turgid, the cell membrane is pressing against the cell wall, there is no space left in between, so it is turgid, right? So if the okay, first of all, let's read further in the question what they are saying. Which row shows the estimated concentrations of the salt solutions in which the samples Y and Z were immersed? So sample Y may be immersed in a solution which is hypertonic, right? Because if it is immersed in a hypertonic solution, if the sample Y would be immersed in a hypertonic solution, then only what will happen, that the water will move out of the cell, and then as the water will move out of the cell, then the cell will become plasmolysed, the cell membrane will be pulled away from the cell wall. So you can say that the position R is basically showing an intense hypertonic solution. And then if I talk about the Z over here, so Z is basically the P over here. How is it the P? Because if I talk about the distilled water over here, so distilled water is a very dilute solution, okay? Distilled water is a very dilute solution. So because of this, what will happen, that as you have placed the sample Z in a distilled water which is a very dilute solution, so let's say that it is a hypotonic solution, so the water will enter the cells by the osmosis and the cell will become turgid and the cell membrane will be pressed against the cell wall. So hence the correct answer over here would be option D. I hope that you guys understood this question. I tried my level best to explain it to you guys. Now let's move on to question number 19 over here. Question number 19 over here says that the graph shows the result of an osmosis investigation using a potato tissue. So they have shown the percentage change in the mass with the concentration of the NaCl sodium chloride. Then further in the question they are saying that what is the concentration of the sodium chloride solution that has the equivalent water potential to the potato tissue, and what is the correct about the movement of water at this point. So when the water potential of the potato tissue will be equal to the water potential of the sodium chloride, then there would be the no net movement of the water, right? When the water potential of the potato tissue will be equal to the water potential of the sodium chloride, so there will external sodium chloride solution, so there will be no net movement of the water, okay? There will be no net movement of water into and out of the cell. And when this will happen when there will be 0% change in the mass. And when is the 0% change in the mass of the potato tissue happening? It is at this point. It is at this point. So at this concentration basically of the sodium chloride, the percentage change of the mass of the potato tissue is 0, which means that at this concentration, the water potential of the potato cell is equal to the water potential of the NaCl solution. So if we read this value, so I believe it is 0.32, 0.34, 36, 37. I believe it is 0.37. So at 0.37 mole per dm cube, the concentration, the water potential of the potato tissue and the sodium chloride solution is same. Now let's read the options one by one. A 0.37 mole per dm cube and no net movement of water. So at what point the water potential is equivalent? So it is at 0.37 mole per dm cube, and over here there is no net movement of the water. So hence we have got our correct answer, which is option A. So I hope that this MCQ is clear. All the other options, you guys can read all the other options. It doesn't make sense. Question 20 over here says that some agar was colored pink using a pH indicator. Then the pink agar was then used to make 3 agar cubes with different dimensions. So they have shown us the 3 agar cubes with different dimension. One is 2, the other is 4, and the third one is 5. The cubes were placed in a beaker and covered with 0.1 mole per dm cube hydrochloric acid. The temperature of the experiment was standardized at 20 degrees Celsius. Hydrochloric acid diffused into the sugar cubes, causing the cubes to become colorless. What is the surface area to volume ratio of the agar cube that becomes colorless in the least amount of time? So basically, the one which will become colorless in the least amount of time would have a greater rate of diffusion, right? In that agar block, there would be a greater rate of diffusion in that agar box, which will become colorless early, right, in the least amount of time. So over there the rate of diffusion would be greater. So the rate of diffusion is greater where the surface area to volume ratio is actually greater. If there is more surface area to volume ratio, then the rate of diffusion also increases. So what we are supposed to do over here, that we are going to calculate the surface area to volume ratio of each of these agar blocks. And how can we calculate the surface area for a cube? Because we know that this is an agar cube. So how can we calculate the surface area for the agar cube? We can calculate it by using the formula 6L square. This is the formula to calculate the surface area for the cube, and for the volume we will use L cube over here. So we are going to put in the values one by one, and then we are going to see. For example, if we calculate the surface area to volume ratio of this, so you can do it with your own calculator as well. You will simply do 6 2 square divided by 2 cube, and then what you are going to get, you will get 3 over here and 1 over here. So for A, let's label it as A. So for A, the surface area to volume ratio is 3:1. For the second block, which is of 4 centimeter, you will get the surface area to be 6, and you can say you will get the volume to be 4, and if you will calculate it, so you will get 1.5:1, right? This is the surface area to volume ratio for the second block. Now coming to the third block surface area will simplify to 6 and the volume will to 5, and then we are going to divide it. We will get 1.2:1. So the correct answer, so basically these are the surface area to volume ratio over here, right? Now the one having the greater surface area to volume ratio will have a greater rate of diffusion, and the one that will have a greater rate of diffusion is that agar cube that will become colorless in the least amount of time. So it would be 3:1, option the agar cube A over here. So the correct answer over here would be option D, 3:1. The agar cube having the surface area to volume ratio of 3:1 will have the least, will show the color, will become colorless in the least amount of time. So that was about question 20. I hope that you guys understood it. So that was it for today's, so that was it for this video. Now we are going to be continuing with question 21 to the question 40 in the other video, which would be the part 2, right? So if you want to watch that part, then I will attach the link of that video in the description box below. You guys can go and check that out as well. Also, I am planning to start my past paper session as well for paper 1 and paper 2, like I did in the May-June series. So if you guys are interested in enrolling in the past paper session, so you guys can contact at this given WhatsApp number. This is the WhatsApp number. You guys can go here, you guys can contact over here. You guys can also contact me through this WhatsApp number, right? So you guys can contact, and we will guide you for the enrollment process in the past paper session, right? Also, if you guys have any confusion in any MCQ, or you have any trouble in understanding any P2 question, you can feel free to send in those questions at the given WhatsApp number, and we will try our level best to guide you guys. Also, I have to tell you about some of the yearly past paper solutions of the paper 2, which I am having with me at my channel as well. So over here in this playlist, you guys can find a lot of yearly past papers, right? Over here I have solved a lot of yearly past papers. You guys can go and check that out as well. Also, I have a topical paper 1 MCQ playlist. This is the playlist for the MCQs, AS level biology topical MCQs. I have covered almost all of the chapters. Some of the chapters I know are left, and I'll cover them very soon. Nucleic acids, mitotic cell cycle, biological molecules, cell structure, enzymes, gas exchange, infectious diseases, immunity. And also I have some yearly past papers of paper 1 solved as well. You guys can go and check that out as well. So this is the past paper solution which I have uploaded. I have uploaded the recent as well: October, November 2024, May, June 2024, October, November 2023. So you guys can go and check that out as well. If you want to enroll in the online classes, you guys can contact at the given number, or you guys can message at this number. And our team will contact you and guide you, right? So that was it for today's class. I hope that you guys understood each and every MCQ I explained. I tried my level best that you guys would understand each and every MCQs. If you guys did and found this video helpful, don't forget to share it with your friends so that they can benefit from this as well. And do remember me in your prayers. Take care. Allah Hafiz everyone.